Hi, everyone. I'd like to thank the organizers of LabRoots for giving me the opportunity to share our research with you today. Before I start talking about our work on antibiotic treatment failure and persistence, I'd just like to acknowledge the students who contributed to the experiments that I'm going to be talking about today. So this is my group here at UConn Health in Farmington, Connecticut, and today's talk will feature experiments that were done by Brandon Bird, Blessing Zenick, Travis Legree, Trish Hare, and Hannah Englander. I'd also like to acknowledge the following funding agencies for their financial support. So, as many of us know, antibiotic treatment failure is imposing a heavy burden on public health systems across the world. And it is projected that this problem is going to intensify in decades to come. And that is why my research lab, like many across the globe, are interested in understanding how bacteria can overcome the effects of our existing antibiotics so that we can preserve the efficacy of our existing drug arsenal and also those are currently in development. So when we think about antibiotic treatment failure, we often think about it in the context of antibiotic resistance. So resistance occurs when a bacterium in a population acquires a heritable genetic change that allows them to grow in the presence of an inhibitory dose of a given antibiotic. So this change may be a point mutation in the primary target of the antibiotic, or the bacterium may acquire a transposable piece of DNA or extra chromosomal piece of DNA like a plasmid that encodes a gene that would allow it to overcome the effects of the drug. So as this resistant mutant grows and divides, then this mutant can pass these genes on to its progeny, spreading resistance in the population. And so in the lab and in many clinical microbiology labs, we can detect resistance development by measuring the minimum inhibitory concentration of a drug for the population. And so, for example, one way of doing this would be doing a broth dilution assay that's illustrated by this cartoon here. So we can try to grow the bacterial strains in increasing concentrations of a given antibiotic, and we can detect resistance emergence by looking at a shift towards higher MIC compared with a wild-type susceptible population. So population-based experiments like these broth dilution assays can tell us a lot about the physiology of a population. So another type of these assays that we may do routinely in the lab is measuring the growth of a bacterial culture, like this growth curve shown here, in which we took E. coli, inoculated it into a nutrient-rich broth, and then monitored the change in optical density in the culture over time. And from the experiment, we might be able to tell that the population initially experiences a lag in growth before it starts to grow exponentially, and then it exhausts nutrients in the population, and then it reaches stationary phase. But what these type of experiments don't tell us is that within that genetically clonal population, there may be bacteria that are behaving differently from one another. So it masks these phenotypic heterogeneity that might occur in the population. So let's take this culture again, and this time, instead of inoculating it in broth, we're going to seed it on agarose pads containing nutrients. So I would like to draw your attention to the 2 E. coli cells that are highlighted by the arrows in the image. And so over time, we can monitor the growth of the population by time-lapse microscopy. And we do this, we see that many of the cells in the population would start to grow and divide, giving rise to many daughter cells by about the 180-minute mark. But we see that the 2 cells that I've indicated with the arrows in the images, they are slower to grow and they hadn't divided within this 3-hour time period. So it indicates that those are slower growing phenotypic variants in the population. So if we were to track the growth of individual cells in the population, looking at how long it takes them to first divide and the length of the cells when they first divide, we see that there is a lot of heterogeneity in the population. And it is thought that phenotypic heterogeneity in the population, whether it is differences in how bacteria grow, their metabolism, and gene expression, those have been linked to their ability to tolerate antibiotics. And so in my lab, we're especially interested in the phenomenon of antibiotic persistence. And antibiotic persistence occurs when you see a genetically clonal culture of bacteria, but within this population, there's a subset of bacteria that may be more tolerant to antibiotics at doses that may kill their genetically clonal kin. So in the lab, we can detect persistence when we take a genetically clonal culture of bacteria and treat it with a high dose of a bactericidal antibiotic, as we depict in panel 2 of this cartoon here. And we track the survivors that remain in the culture and plot the survival along the y-axis of this curve on a log scale and time on a linear scale along the x-axis, we may typically see biphasic killing, as you can see in panel 2 here. So what this tells us is that this population is phenotypically heterogeneous, and it consists of a population that may be rapidly killed by the drug, as you can see in the first phase of this kill curve, but there is also a subpopulation of cells, point persisters, that may be killed at a slower rate or not at all. So you take the persisters that remain at the end of this course of treatment, dilute the drug to sub-MIC levels, and then allow the persisters to recover and resume growth in the presence of nutrients. And then you take the resulting population and treat it with the same antibiotic again. You would typically see biphasic killing again. And oftentimes the slopes of the kill curve may be similar to what you see in the original kill curve. So this indicates that the persister phenotype is not heritable. And when persisters resume growth, they can grow and give rise to both persisters and non-persisters. So this is a cartoon depiction of persistence. In the lab, we certainly see persisters in a number of different bacterial species. So in my lab, we generally study persistence in E. coli, Pseudomonas aeruginosa, and also in Staphylococcus aureus, but persistence has been reported in a number of different bacterial pathogens. So if we take a population of E. coli that's susceptible to this antibiotic, which is a fluoroquinolone called levofloxacin, this population is susceptible to the drug, and the MIC is 0.023 micrograms per mil. But if we take this culture and allow it to grow and then treat the population with a high dose of levofloxacin, so here we had applied 5 micrograms per mil of levofloxacin. So this far exceeds 100 times above the MIC. And we track the survivors that remain over time. We see this biphasic kill curve, indicating that there are persisters in this culture. So even though the persister phenotype is not heritable, persistence has been thought to underlie relapsing infections. And as you will see in a few slides, our group and others have shown that persistence can accelerate the development of antibiotic resistance. And that is why my group and many others are interested in understanding how persisters can overcome the effects of our existing antibiotics. So in my lab, we're especially interested in understanding how persisters can survive treatment with topoisomerase inhibitors. And these drugs target topoisomerases like DNA gyrases, which can change the supercoiling state of DNA during DNA replication and transcription. And at high concentrations, these drugs can lead to DNA double-stranded breaks and cell death. So if we treat populations of E. coli with high doses of fluoroquinolones, that is one of these topoisomerase inhibitors, so treating them with a drug like levofloxacin, we can see that they can kill these populations. And we also see that when we treat a population that is growing in the presence of glucose, this drug can reduce the survival by about 3 to 4 orders of magnitude. But if we take a population that has been starved of glucose, and so they're in a slower or non-growing state, as you can see in the red curve there, we see that the drug can still kill about 90% of the population, but the survival and persister levels is much higher compared with what we had seen in the growing culture that was in a glucose-rich environment. So we're especially interested in understanding how bacterial populations that are slower and non-growing can respond to antibiotics, especially how persisters in these cultures can survive, because these cultures are generally more tolerant to a range of different antibiotics that we currently have on hand. So some of the questions that we set out to ask when we first designed this project include, so we see that these persisters can survive treatment with lethal doses of these topoisomerase inhibitors. So do these persisters still suffer from DNA damage? What defenses do these persisters depend on to survive treatment with these topoisomerase inhibitors? And can we leverage what we learn about how they survive existing topoisomerase inhibitors to try to potentiate the activity and sensitize these persisters to new topoisomerase inhibitors that are in clinical development? So in answer to our first question, do these persisters that can survive topoisomerase inhibitor treatment still suffer from DNA damage? We took on a relatively simple genetic approach in which we deleted a gene called REC-A, which is a key player in the DNA double-stranded break repair and also in orchestrating the DNA damage repair SOS response in bacteria like E. coli. And we see that in this mutant, the survival of the population was treated with levofloxacin, a fluoroquinolone, decreases by about 3 to 4 orders of magnitude compared to its wild-type counterpart. So this result suggests that these persisters do indeed suffer from DNA damage, and they depend on RECA to survive. So to look at when these persisters may be repairing their damaged DNA, we made use of a fluorescent gene expression reporter that reports on the expression of RECA. So in these plasmid-borne reporters, the promoter of RECA is transcriptionally fused to a gene encoding the green fluorescent protein, or GFP. So we can transform these plasmids into our bacteria and then look at when fluorescence is induced to gauge when RECA may be induced. So first, we treated a population of slow or non-growing culture of E. coli with levofloxacin. And after a period of treatment that would leave persisters as the sole culturable cells in the population, we diluted the antibiotics sub-MIC levels and allowed the cells to recover in the presence of nutrients. And we monitored the expression of this RECA reporter over time. And we did this, we saw that when these cells were first inoculated in this antibiotic-free media containing nutrients, most of the cells remained dim. And it wasn't until they had time to recover in this media that we see by flow cytometry that there's a shift toward higher fluorescence in about 90% of the population. So this suggests that maybe the cells were activating RECA expression as they're recovering after fluoroquinolone treatment. So to look at this further, we inoculated these cells on agarose pads containing nutrients as they recovered from antibiotic treatment. And this way we can monitor these cells by time-lapse fluorescence microscopy. And consistent with our flow cytometry histograms, we see in these images that most of these cells remain dim when they were first inoculated onto these agarose pads. And as they had time to recover in the presence of nutrients when the drug has been diluted to sub-MIC levels, we see that the majority of cells in the population fluoresced, and they also filamented, as you can see in the image in the 432-minute mark. And so they fluoresced and filamented regardless of whether they go on to live or to die. And so they form these long filaments because one of the genes that is part of the SOS DNA damage response is SOL-A. And SOL-A inhibits cell division until the cells have had adequate time to repair damaged DNA. So we also see that in these images, there's one cell that is indicated by the red arrow that at some point around the 468-minute mark, it splits into two. And then it further divides, giving rise to a microcolony by the 540-minute mark, and it dilutes the fluorescence as it divides. So that is an example of a persister. So collectively, these data suggests that in persisters originating from slow or non-growing E. coli cultures that have been treated with fluoroquinolones, these cells still suffer from DNA damage, and they depend on DNA damage repair in the post-antibiotic treatment recovery period to survive treatment. So we then asked, what other defenses do these cells depend on to survive treatment with these fluoroquinolones? So to address this, we turned to genes that encode these multi-drug efflux pumps, including the resistance nodulation division family of pumps, including ACR-AB and TOL-C in E. coli. So this work was led by a former MD PhD student, Brandon Bird, together with a former postbac student, Blessing Zenick. And we decided to look at the contributions of these pumps because fluoroquinolones like levofloxacin and a new fourth generation fluoroquinolone called Delafloxacin, they're substrates of these multi-drug efflux pumps. And so if we're to delete genes encoding components of these pumps, like deleting ACRB or TOL-C, we see that the MICs of these drugs decreased considerably compared with the wild-type counterparts. So we then asked, because these drugs are substrates of this pump, how does loss of genes encoding the pump components affect persistence of the mutants when they're treated with these drugs when they're in a slow or non-growing state? And interestingly, we saw that even though levofloxacin is a substrate of the pump, loss of pump action did not affect persistence toward levofloxacin. But on the other hand, we saw that in populations treated with Delafloxacin, loss of ACRB, and we had also deleted TOL-C, that resulted in a decrease in survival of about 10 to 100-fold compared to the wild-type cells. So we then asked, what happens to the other fluoroquinolones that are also substrates of these pumps? Does the loss of pump action affect their persistence like what we saw with Delafloxacin, or is it more like what we had observed with Levofloxacin? So we also treated these populations with two other fluoroquinolones, ciprofloxacin and clinofloxacin. And in both of these treatments, the phenotype that we observed was reminiscent of what we had seen with populations treated with levofloxacin. So loss of pump action did not affect persistence toward these two fluoroquinolones. So these data indicate that loss of efflux pump action only really affected persistence toward Delafloxacin in these slow or non-growing populations of E. coli. So previously it's been shown that bacterial persisters may use different strategies and defenses to overcome the effects of different classes of antibiotics that target distinct cellular processes. So here what we're seeing is that perhaps for cells treated with different compounds within the same class of drugs, depending on the compound that is used, cells may also mount different defenses to survive treatment with the drug. So if we look at the structures of these different fluoroquinolones, we see that levofloxacin, ciprofloxacin, and clinofloxacin, there's zwitterionic, a neutral pH, whereas delafloxacin is anionic. So it is thought that delafloxacin may be able to better accumulate inside cells, and that might explain why these persisters depend on efflux pumps to overcome the effects of delafloxacin. So as I had mentioned, when fluoroquinolone persisters originating from slow or non-growing cultures are treated with the antibiotics, they suffer from DNA damage, and they need to undergo DNA damage repair after antibiotic treatment is over to survive. So here we wanted to ask, do these delafloxacin persisters depend on the effects of the efflux pumps during delafloxacin treatment or once treatment is over? So to address this, we made use of a small molecule competitor for the efflux pump called PA-beta-N. And so with this competitor, we can administer it either together with delafloxacin, so disrupting pump action during delafloxacin treatment, or we would add PA-beta-N to the cultures during the post-antibiotic treatment recovery period. So once delafloxacin has been diluted to sub-MIC levels and cells are recovering in the presence of nutrients. And so when we carried this out, we saw that when PA-beta-N is added together with delafloxacin, as you can see in the red curves here, survival of the population decreased by about 10 to 100-fold compared with cells that were treated with delafloxacin alone. And this is similar to what we had observed with the genetic mutants that you had seen a few slides ago. And when PA-beta-N is added after delafloxacin treatment is over, we saw that the survival of the population is comparable to cells that were treated with Dela alone, as you can see in the black and gray curves in the graph. So this demonstrates that these delafloxacin persisters from these slow and non-growing populations were dependent on efflux pump action during delafloxacin treatment. So previously, it's been shown that when fluoroquinolone persisters originating from slow or non-growing cultures are treated with the drug, and then the persister is allowed to recover in the presence of nutrients once the drug is removed, when you challenge the progenies of these persisters with different antibiotics, you would detect an increase in resistance development in the persister progeny compared with populations that were not exposed to the fluoroquinolones. And here, what we had seen was that we observed an increase in the number of resistant mutants in the persister progenies, not only when these cells were treated with the fluoroquinolone that was originally added to the cultures, so ofloxacin in this case, we also saw an increase in the number of resistant mutants in the persister progenies when they were exposed to rifampicin, which targets RNA polymerases, and also when these progenies of persisters were exposed to a number of different cell wall synthesis inhibitor, like carbenicillin, D-cycloserine, and fosfomycin. So here we wanted to ask whether these delafloxacin persister progenies also exhibit an increase in the number of resistant mutants in the population? And how does the disruption of these multi-drug efflux pumps during Delafloxacin treatment affect resistance development in the Persister progenies? So first, we had seen that when these cells are exposed to Delafloxacin and the persisters are given the opportunity to recover from treatment, we see that there is an increase in the number of rifampicin-resistant mutant in the persister-derived cells, similar to what we had seen in populations that were treated with levofloxacin and other fluoroquinolone. And as I had mentioned, for levofloxacin persisters, we had seen that loss of efflux pump action during levo treatment did not affect persistence toward the drug. And we also saw that loss of efflux pump action by deleting ACRB did not affect resistance development in the levo persister progeny. But we had seen that for cells that were treated with Delafloxacin, loss of efflux pump action either by deleting ACRB or Tol-C or exposing the cells to PA-beta-N during Delafloxacin treatment, that resulted in a reduction in resistance development in the Dela persister progeny. And it wasn't until we had complemented ACRB back into the deletion mutant that we saw an increase in the number of resistant mutant, similar to what we had seen in the wild-type population. So these data suggest that perhaps disrupting efflux pump action during Delafloxacin treatment could affect resistance development in the persister progenies, and maybe disrupting efflux pump action during treatment could be affecting the coordination of molecular events during the post-antibiotic treatment recovery period as these cells are repairing DNA and resuscitating after antibiotic treatment. So right now, my group is investigating this to get a better understanding of what is going on. So from this part of the talk, I hope I've shown you that for slow or non-growing populations of E. coli that are treated with older generations of fluoroquinolones like levofloxacin, Cipro, and clinofloxacin, loss of efflux pump action did not affect persistence toward these cells. But for populations treated with Delafloxacin, disruption of multi-drug efflux pumps like ACRAB and Tol-C could decrease persistence toward Delafloxacin. And that could also affect resistance development, reducing resistance development in the progenies of these Delafloxacin persisters. So next, we wanted to see if we can take what we had learned about some of the defenses and vulnerabilities of these fluoroquinolone persisters to try to sensitize slow and non-growing populations of bacteria like E. coli to new non-fluoroquinolone topoisomerase inhibitors. And this work was once again led by Brandon together with PhD student Travis Legree. And we decided to focus on Zoliflodacin, which is currently in phase 3 clinical trials. So because Zoliflodacin is a topoisomerase inhibitor, we first asked whether Zoliflodacin, like what we had seen with fluoroquinolones, whether it can also kill slow or non-growing populations of E. coli. And the answer we got back was no. So we had applied Zoliflodacin at high doses at about 100 micrograms per mil, which is about 50 times above the MIC, and we didn't see an effect on the survival of these populations. So Zoliflodacin is a substrate of ACRAB Tol-C, and so we asked, how does the loss of efflux pump action affect Zoliflodacin? Do we see a reduction in survival similar to what we had seen with Delafloxacin? And we saw that loss of ACRB did not affect persistence towards Zoliflodacin. So previously, it has been demonstrated that bacterial persisters and cells that are in a slow or non-growing stage, they can still retain metabolic capacity, and they can respond to metabolic stimulation, which can sensitize them to different classes of antibiotics. And these include aminoglycosides that target the ribosome, fluoroquinolones, and nitrofurans. So Travis and Brandon then looked at whether they can stimulate the metabolism of these slow and non-growing populations of E. coli with glucose, and whether that would sensitize them to Zoliflodacin. And they saw that at the highest concentration that they tested, adding glucose to stimulate the metabolism of the cells did significantly decrease the survival of the population by about 10-fold. And it wasn't until we combined the effects of metabolic stimulation together with loss of efflux pump action, as you can see by the purple curve, that we saw a significant decrease in survival of these slower non-grown populations treated with zoliflodacin across a range of Zoli concentrations that we had tested. So this demonstrates that a spoonful of sugar combined with a knock to the multi-drug efflux pumps of these slow and non-growing population of bacteria could help sensitize them to zoliflodacin. So we then asked whether these zoliflodacin sensitized cells still suffer from DNA damage, and once again we made use of our delta REC-A mutant, and we saw that when cells lacked REC-A, survival of these cells decreased by about 5 orders of magnitude compared with their wild-type counterparts when they're sensitized to zoliflodacin. So this indicates that these persisters that survive Zoli treatment suffer from DNA damage. And to look at when these persisters may be recovering and repairing damaged DNA, we made use of an inducible copy of REC-A and complemented the delta REC-A mutant with this construct. So with this construct, what we're able to do is to tinker with the timing of REC-A expression. So either inducing REC-A expression before, during, or after zoliflodacin treatment. And so we can look at when REC-A expression may be required. And so when we did this, we saw that regardless of whether REC-A was induced throughout the course of zoliflodacin treatment, so as you can see in conditions 1 and 2, or whether REC-A was only expressed during the post-antibiotic treatment recovery period, as you can see in condition 3, survival of the populations were comparable. So this suggests that these cells really only depended on DNA damage repair and REC-A action as they're recovering from zoliflodacin treatment. So this suggests that maybe there is a window of opportunity for us to target these cells that have survived fluoroquinolone and zoliflodacin treatment to try to reduce the burden of persisters in the post-antibiotic treatment recovery period. So to look into this, we decided to take on a proof of concept experiment to see if we can leverage microbial competition to try to kill persisters as they're recovering from treatment. And this work was led by DMD PhD student Trish Hare and a former undergraduate student, Hannah Englander. And so Trish and Hannah decided to focus on E. coli Nissl, which is a probiotic strain of E. coli that is an active ingredient in a product that's marketed as Mutaflor in parts of Europe and in Canada. So Trish and Hannah took a slower non-growing population of E. coli and treated it with levofloxacin, and then they allowed the persisters to recover in lysogeny broth, either together with a lab strain of E. coli or together with E. coli Nissl. And they monitored the recovery of these levofloxacin-treated cells over time. And they saw that when E. coli Nissl is added at a concentration that is about 10 to 100-fold below the density of the levofloxacin-treated cells, E. coli Nissl, as you can see in the red curve of the triangle and the circles, they could suppress the recovery of the levofloxacin-treated cells and reduce persistence in the culture. So to look at how E. coli Nissl is doing this, we first asked if Nissl is acting in a contact-dependent or contact-independent manner. And so to address this question, we made use of a device that is custom-made that we call the H cell, because it looks like an electrochemical H cell. And this would allow us to culture E. coli Nissl and the levofloxacin-treated cells in separate chambers that's separated by a filter that would allow small molecules and nutrients to pass through, but not the cells. And we saw that when E. coli Nissl was cultured with the levofloxacin-treated cells in this manner, Nissl was no longer able to inhibit the recovery and reduce the burden of the levopersisters. So this suggests that the effects of E. coli Nissl is dependent on either direct cell-cell contact or Nissl needs to be in close proximity with the levofloxacin-treated cells for it to exert its effect. And so right now, our lab is trying to understand and elucidate the molecular basis of these interactions. So I hope I have shown you in today's talk that we see that persisters that can survive fluoroquinolone and topoisomerase inhibitor treatments when they're in a slower non-growing state, they still suffer from DNA damage and they need to undergo DNA damage repair in the post-antibiotic treatment recovery period to survive. And so we can try to leverage approaches like using microbial competition to reduce the burden of persisters during this period. We also see that depending on the drug that is used, Some of these persisters may depend on multi-drug efflux pumps to survive treatment with these antibiotics, as we had seen with Delafloxacin. And so understanding the dependence of these multi-drug efflux pumps, we showed that we can inhibit efflux pump action together with stimulating the metabolism of these slower non-growing populations of E. coli to sensitize them to a new non-fluoroquinolone topoisomerase inhibitor called Zoliflodacin. And so I hope I've shown you today that there are ways for us to leverage what we learn about the vulnerability and defenses of these persisters to try to sensitize them to existing and emerging antibiotics. And so with that, I will end here, and I would be happy to take questions offline. And thanks again for tuning into this webinar.